I mean,
And
I don't. I thought about texting you earlier. Did I send you a text about potentially us, not limiting ourselves to this paper or talking about something else? It's not.
I flatly rejected it.
Yeah, there was no response for sure.
Right.
I think they're based on yours and my one-on-one conversations. I think there's some interesting stuff, and it will certainly entice the Brotherhood to respond.
The Brotherhood. Okay. So I'm going to share a screen, but if we can work out, have you worked out any of the math here?
Not personally. Beyond the understanding that it takes the basic Shannon entropy equation, which I'm going to pull up, and it derives. There's like 3 or 4 levels of derivation.
And the interesting thing is, like, he shows you how to do Shannon information across one genome, and then he goes to, okay, but that's really useless. That just tells you how variable, how much it diverges from random. So, which doesn't give you any information at all. I mean, that information would be meaningful if you had to transfer a genome over a wire, or if you had to send it to somebody, right? Well, or copy it, and may make a difference.
That's what he's saying, is that effectively, and he says this, he states it very well, but effectively, if you apply conventional Shannon entropy theory and equations to genetics, that when in a sequence, when you come to a start codon or promoter sequences, that if you base it entirely upon just kind of basic information theory, there's no reason to expect an open reading frame to follow the promoter sequence. I think that's one of his level of derivation that we see in this paper. Ultimately, it seems like he concludes.
Oh, did he cut out?
We might have lost him for a moment.
That we don't need to be thinking of this as information, but as what he calls biological meaning, and the best way to quantify meaning.
Oh, you're breaking up real bad.
Can you guys hear me?
Yeah, you cut out after you said ultimately he concludes. So
I don't
the
know.
last 30 seconds or so.
Yeah.
Gone out.
Then we lost you.
Damn internet.
Yep. It sucks. Oh, okay.
How about now?
Yeah, you're doing
ultimately he concludes that the best way to identify and quantify biological meaning, the best way to identify biological meaning is to look across species. So to look for sequence homology. That basically the meaning that you find is a roadmap of all the selective pressures that lineage has gone through, over the millennia.
Yeah, and that's what's highlighted.
Not just look at it as though it's an output.
Mm-hmm. That's what's highlighted here in the abstract is vertically but not horizontally. So he looks at Shannon information vertically across different species instead of horizontally across the genome, which would be fairly not exactly meaningful information.
I think.
So he's
was horizontal where you look at like the sequence and then the function, versus vertical, like you were saying, homology between species.
No, it's actually like
horizontal is like gene expression.
Nope, it's actually even
he
less
chose
than
not
that.
To go.
It's even less meaningful than that, and he's basically looking at the, let's see, applied this way, entropy only tells us how far the count of each base deviates from the expected sequence, which does not convey any meaningful message on the genome in question, let alone the phenotype of the organism. So in other words, applying Shannon information this way would be useless. So that's kind of what he's saying. So
yeah, and that's if you Google Shannon information or Shannon entropy equation, that's basically it. Some of the variables, like the letter representations, are different.
And it's just
that's what the argument he and Ademi, if I'm not butchering the name, the argument they make is that you can't look at biological information using standard information theory.
Yep, it's the Italian is Adami or Adami. So
Adami.
Yeah, there's a little pronunciation thing on the internet that you can get that's pretty helpful, but they'll do it in like the Italian form. So Adami, I don't know. Okay, so that's useless. So the next thing he suggests is that let's see. In qualitative terms, biological information is perhaps best described as the meaning of a sequence. A nucleotide sequence assumes meaning only when it is either transcribed into a protein or an RNA molecule that directly carries out a biological function. So it would have to be something that makes a difference in the cell. And then he goes on to...
Not like telomeres and centromeres, not regions of chromosomes that are tightly compacted and have very little transcription binding. And
arguably those could make a difference too, but they're more mechanical, like the length of the genome in a particular organization or organism would be more specific to that organism. So it's kind of hard to make that comparison. Well, we haven't gone there yet. So he's talking about trying to figure out how we would make meaning within an organism. And what you just brought up is not addressed in this paper, but perhaps it should be. Like centromeres are important. The length of the genome when it arranges itself around the nucleus, the 4D nucleome, and how things come into play with epigenetics may all be useful. So there's more that can be added, but he kind of focuses on like RNA transcripts. So RNA transcripts, whether they be ones that create proteins or ones that create regulatory functions.
It does make a certain amount of sense. If he's looking for meaning, if he's looking for the interplay between the genome and the environment over successive generations, it's going to be a much more direct relationship between, gene loci than presumably than telomeres or centromeres, which understandably would be more static or less flexible or responsive to selective pressures.
Or long areas of tandem repeats, be it from retrotransposons or transposons or just genetic copy errors. So those long areas of repeats would be hard to measure across an organism. But first of all, he says, okay, what you could do in order to find meaning in a cell is like what we were just talking about. You take each piece of it and do an exhaustive characterization of the biological role of each nucleotide in the genome is unrealistic for even the smallest genomes. So we're working on that. We're working on figuring out what everything does, but we don't know exactly. Standing for Truth often says the gold standard of science or genetics is to do knockout experiments. That's not true at all. The gold standard is figuring out what each of these sections do, what they're but it's impossible to do that.
I can kind of understand that statement if it was more qualified. As far as linking a gene to a particular phenotype, or certainly determining if a gene is essential.
Yeah, that doesn't tell you the whole.
Knockout experiments will answer that question. But you're absolutely right. a full characterization involves, tissue expression patterns, response to various signaling pathways. There's a whole lot more that goes into it beyond knocking out the gene. That just tells you whether or not it's essential for the organism to live.
Yep. So Walker, what do you think? You follow so far? We're basically trying to figure out what the information is. Do you have any comments?
Well, I mean, right now we're just in the abstract, where he's
actually I'm on page.
Ideas.
I'm on page 1.
Yeah, that's what I was saying. Like, this is still abstract, right? No. Oh, okay.
It's abstract in the sense that it's the introduction.
Yeah, it's introduction. So this is section 1, entropy information, meaning and genome evolution. So, okay, that's the first part. So we're on page 1. So try to keep up.
Yeah.
I'm sorry.
So I don't know if Walker knew this, but when Speed and I talked about this previously, I pointed out that this was one of like 23 papers published by the Royal Society. I think was it 2016. I think this.
Yes, it is. Yeah, and I'm going to show that page in the intro. And I'll probably
it's a thematic volume. I think 337. I can't remember the specifics, but the theme of all the papers, 20-something papers, is DNA as information. So they have a multidisciplinary team, engineers, computer guys, biochemists, molecular biologists, and they're all kind of weighing in on this principle of DNA is an information-rich substance. It's actually a side of molecular biology that's taken for granted from my position. We use these genes. We use we have a whole toolbox of techniques, and we can do all kinds of crazy stuff in the lab. But this is a little bit more philosophical, but backed up by this approach to actually quantify biological meaning, which I think will build to this first figure. I'll hand it back over to Speed.
Yeah, well, let's go ahead and take a look at that. Is this visible on your screens?
Yes.
Fine?
You're good.
Could I make this bigger? Or it doesn't really make any difference. So we'll do it this way. Okay, so this is the Royal Society journal issue that John Maddox in particular referred to very early on when he came onto the scene. And this is the theme issue, DNA as Information, compiled and edited by Cartwright and Giannarini and Gonzalez. So we have the introduction, which is something that would be worth looking at some point, maybe even earlier. And then the first paper is the meaning of biological information, which is what we're looking at. Well, then we've got Roederer, which is a very interesting paper where he talks about pragmatic information, and he expands on this one a little bit. And let's see how much it has been 5 or 6 months since I read these papers, but then Varn and Crutchfield, what did Irwin mean? We've got the information architecture of the cell. Here's Christoph Adami with what is information, and that's probably the next one we're going to address.
That's a really good one, and it's a really easy read because it's like he's having a conversation with you, and so just the style and the format he kind of writes it in is like a conversation.
He does a real good job of explaining entropy, I think, in a way that you can intuitively understand.
Like Shannon entropy? Information theory entropy, or
information theory. Like he defines it as uncertainty. Like if you're going to roll a die, you have what is it? Your maximum uncertainty, because you don't know which of the 6 states it's going to result in. And then he goes on to explain that you could be looking at something else instead of just which number comes up. You could be looking at, is it going to be even or odd? Well, that changes the amount of entropy. So So a measure of entropy is dependent on what you want out of the measurement. And he makes that case, which is pretty cool. But again, it's been, we have to go over that paper. It has been a while, but it's very interesting how he intuitively defines information. I think he's the one that takes a good shot at pragmatic information, though it may be Roederer. Well, then we have, what else we got? The Generation of Meaningful Information in Molecular Systems by Wills. And this is a really good one, Wills paper. Let's see. Self-referring DNA and protein. A remark on the physical and geometrical aspects. I think that, that may be remarking on what we were talking about with centromeres and length. That could be the 4D nucleotomes. Then we have a little bit of graph theory. I mean, there are just many papers here to go over. What is Information by Barbieri is one I believe that the Maddox refers to directly. I'm not sure where, but he may. He doesn't even show you these papers other than a brief glance, so it's a good idea. But this is a really good one. What is it? Then Problems of Biological Information by Ball, another good one. Got another one by Wills. He was busy. This one haven't looked at. So there's just a whole. I'm not sure how many. I thought there were about 15 or 16 papers. I'm not sure. So anyway,
I thought it said it was like 22 or 23 in that under that theme of DNA as information.
Right. And this has been described as setting biology on its ear. Well, that happened actually starting in about 1986, where they had that conference, and they ended up the information theorists and the dynamicists, I call them, looking at biology as a dynamical system. They started pushing back on some of the just standard ideas about biology and looking at it more as dynamical systems. And they were also looking at ecologies at the time as dynamical systems with feedback. So this whole, so there's been this sort of brewing war where you'll have one session that'll say it's just chemistry, and you'll have another session say, no, the overall dynamicism and information is important too. And then it kind of came up in around 2010. Some good material started appearing everywhere about all of this, including the biosemiotics book and all of that. And there's a book here by Jesper Hoffmeyer called Biosemiotics, an examination into the signs of life and the life of signs. So that's a good one to give us some information going forward. But so anyway, that's the Royal Society. Is anybody have comment on that?
Is the paper we're reading right now also one that Maddox cites, or does he just cite this whole volume?
He has cited a paper by Koonin, but a different one, and it's not in this volume. So the paper he cited is, let me see here.
You digging through your Maddox stack?
I am, and I shouldn't be doing that.
Koonin is prolific, though. I mean, he's written a ton of stuff.
Okay, the one that Maddox cites in his the last talk that he did, he pops it up for just a minute. The Origin and Evolution of Genetic Code, the Universal Enigma, and he liked it because it says enigma. So sure. That's a Koonin 2009. That was prior to this particular volume. So a little more dated, but also it wasn't a particularly rich paper. It just went on and talked about the DNA as a code and the conundrum. There are some interesting things in there. We will, I think, go over that. There are things in there about why the code is what the code is. And I think he says that it is not the most efficient or least error-prone code that could have been chosen. He says that it is somewhat less than completely optimum, and they've shown this in a number of different ways, but it is a pretty damn good one. It was selected for its lack of errors, and I believe for specificity or whatever. I'm not entirely. It's a good code out of the 10 to the 84th possible codes that could have been chosen. So.
it sounds like everything in biology, you know, good enough is perfect.
Yeah. The case has been made that it's the most perfect code. Well, there's people pushing back on that. They've mathematically determined could have been better. Yeah. But well, you know, toward the sixth day, God was getting a little tired. And he
Well, I
probably
mean, at this point, he's already created. Like the genetic code would have had to come about on the third day or on the fourth day, because that's when the sea creatures, the birds, are.
Oh, that's true.
created.
Well, hell, then he has no excuse for what he did. Yeah.
Are you gonna pull up the Koonin paper again?
I am.
I liked you were going through and reading the areas that you highlighted. I like that. I think it does a good job of kind of staying true to Koonin's, you know, claims, but also not being so dense that it's hard to follow.
Yep. And now, of course, StreamYard lost it. So fortunately, I can cut we can say anything in between. You make a note or whatever. I can cut all this stuff out. It's going to take a little editing, though.
The question is, will you?
Oh, I know. That is a very valid point, Walker. It's a lot of work. And I just have to stay undistracted. So, okay. This really irritates me that StreamYard does this. You have to click on it, and you click on it a couple of times and then pull it up. And then StreamYard hopefully finds it. Okay. I was looking in the wrong place. It wasn't StreamYard's fault. It was mine. But I'm not going to admit that I'm wrong live. I'm cutting this part out.
So in the section that we're in is basically Koonin with Adami, and he cites Adami throughout this paper. He's building the argument that the most accurate or the best way to look at information in DNA is what he calls biological meaning.
Yep.
And it builds up to this. It's not the basic Shannon entropy equation, but it's a derivation of that takes into account kind of the specific characteristics of DNA that Speed was talking about earlier. You know, that certain sequences are associated with specific biological functions, which ultimately are associated with morphology, physiology, phenotype.
Yeah, I think he goes through that in the next paragraphs. He talks about delineating regions that are most likely used to produce proteins. The regulatory they contribute to biological function by purifying selection. Okay, now he's talking about like if we were to look at a phylogeny and we were to look at primates, then within primates there would be a certain subset of our genome. And it isn't that great a percentage that is completely conserved. And then if you look deeper, like you look at all mammals, you'll find a smaller set that's completely conserved, I would expect. And then you look all the way down to simple eukaryotic cells, fungi, whatever. You're going to start to find the meaning in the metabolome, the things that are really important for a cell to create its energy and work at all for a eukaryotic. And then go down to proteomes and right down to the simplest cell, and you'll find a difference, I think, depending on which cut you make. You're going to find a difference in this bio. Is that correct? That you would find a difference in biological information?
Well,
between these,
that's what he's building to. So maybe we should. But this section that you're kind of scrolling through here, he says, however, the only general way to extract meaning from sequences involves comparative analysis of homologs. The premises are extremely simple yet powerful. The great majority of the meaningful sites in nucleotide sequences, i.e. those sites that contribute to biological function, are subject to purifying selection, hence evolutionary conservation of meaningful sites. So here's the take home. The stronger the selection, the more meaningful or important a site is. So and above, so this is the vertical analysis versus horizontal. So what you covered earlier, Speed, what he was talking about above, where you look at, what biological function does a particular sequence have. That's the horizontal, approach. But
Yeah.
he's saying if you want to look at meaning, i.e., what is most evolutionarily conserved, you use, I think, equation 1.4, 1.5. We don't have to get too deep into the math, because the value of the math is kind of intuitively obvious once you see the first figure.
Mhm.
Yep. Yeah, so he's just looking at basically entropy vertically across organisms rather than entropy horizontally across one particular genome, which would just be, how much noise is in or how much, basically how far it deviates from random vertically, which makes no sense. So anyway, so we've gotten that far, and the math does not really make a difference. The thing we have to understand is how he comes up with this information density. And I think, does he derive that here? The length of the genome. Okay, hang on a second. Biological evolutionary information seems to be a more straightforward definition. So that's what he's striving for. And now the interesting thing is that creationists often ask you, or they often try to frame, they will say that no new information is gained, right?
Mm-hmm.
And this is kind of plays against that. So biological information
on 2 levels. It plays against it on 2 levels, which we'll see with figure 1.
Okay. Let's see. And then thus, Dobzhansky's famous dictum, nothing in biology makes sense except in the light of evolution, which is kind of what he's saying, that in order to figure out what is important in biology, I mean, this kind of emphasizes that you have to look vertically and you have to look at it as having evolved in order to make these determinations, I think, is what he's saying. So. Okay. So where are we here? To conclude this conceptual discussion, seems pertinent to ask, what is biological information about? Oh, let's see here. This is really important. It has been persuasively argued that biological information stores the history of the environment that an organism has been in, information about the environment, allowing the organism to predict and exploit environmental changes, and also sort of records the entire history of the evolution of that organism. So in a way, we're recording history.
It literally records it, as we see with these phylogeny trees. But he's saying there's a deeper record of meaning that's also recorded.
Yep. Let's see, it's really important.
Do you remember where the provocative statement was?
I think it's right after the graph.
If you want to jump, some of this stuff can get really dry. Speed. Okay. I think it's that.
Am I boring you now?
Not at all. Not at all.
Oh, that's okay. Let's see. Where are we here? That's the genomes of organisms. I'm trying to find.
I think that's the quote he was talking about right there. Yeah.
This one?
Yeah.
Yeah. Okay. So somebody want to read that?
Go ahead, Walker.
All right. Thus, the genomes of organisms that are usually perceived as the most complex, such as animals and plants, indeed have the highest total information content, but are also entropic genomes with low biological information density. By contrast, organisms that we traditionally think of as primitive, such as bacteria, have informational genomes with high information density. To rephrase the same statement more provocatively, the genomes of unicellular organisms and viruses appear to be incomparably better designed than the genomes of plants and particularly animals.
That's a pretty stunning statement to find, and I'm guessing that no creationist would ever highlight this one in these papers.
Yeah.
I'm guessing Maddox would never. I'm guessing if Maddox did read that, it didn't record at all.
I doubt he read it.
Yeah, I doubt it. But if he did, all you would hear is that wah 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 wah. That we hear with Charlie Brown.
I anticipate it could probably be rationalized by defining or by appealing to divine mystery or something to that effect.
Yeah.
Not that that's really a rationalization, but
yeah. So basically, in simpler organisms are wall-to-wall genomes. I got to learn to pronounce things the same as you guys. That's what happens when you read by yourself.
sure.
Oh,
Yeah.
For sure. I mostly realized that with like talking about fossil taxa, but carry on.
Yeah. Yep. They have different ways of pronouncing it. So the wall-to-wall genomes you'll find in simpler bacteria and things like that, and that's they're simpler, they're more direct, they work just as well. They have similar mutation rates overall. When you consider the rapidity, how rapidly they multiply, and they seem to work just as well with wall-to-wall, completely packed genomes.
Mm.
Whereas we have a lot of strings in between that are suspicious to me. I don't know. Anyway.
So looking down at this next part, which mentions the ENCODE project, but
Yes.
Can we look at Figure 1 before we jump into that? Because I like that section about fuzzy meaning, and actually rereading it, it was pretty interesting. But
It is.
But after what you just stated about, or what Walker just read, this is the take home. So I can walk you guys through this figure if you want, or speed, by all means, you can as well.
No, we most certainly want you to do it.
Okay, so there are a couple of different vectors going on with this chart. So to avoid getting into the mathematics, let me start with kind of the simplest way to look at it. If you see that there are either red squares or blue diamonds representing kind of what the two icons or symbols represent is the two different ways to look at information in genomes. So going back to something Speed said earlier about kind of the creationist take on no new information added, like literally from the blue diamonds represent, the number 1 is the simplest, most primitive form of organism, up to number 11, which is humans. Number 10 is the fruit fly. So literally there's a spectrum of information that has been added. Actually, we may not want to overstate this. I don't want to overstate this, because effectively what we're looking at is extant organisms.
Mm.
But if you are convinced by the fossil record and by evolutionary models, 1, 2, and 3 represent organisms at the earliest stages of the development of life. Before multicellularity, number 1's before even.
Well, 1, 2, and 3 are all actually viruses. It's RNA viruses and then 2 phages.
So it's even preceding.
Like,
Well, yeah. Let me not state that, because it's arguable whether viruses preceded cells, or if viruses are actually derived from cells. There's good evidence for the latter. But basically, so I don't know if you want to edit all that out, but I can actually start over. So in Figure 1 here, we see Koonin plotting out the two ways to look at information. So the blue diamonds just represent total information content. So 1, 2, and 3 are viral genomes, and you effectively as you go up, you increase complexity and also increase total size. So if intuitively you would expect a animal to have more information in its genome than a bacteria. And this is the pattern that you see. 11 is the human. We can see the blue diamond in the top right. Number 10 is the fruit fly. Also, lots of information in its genome.
Mhm.
But what Koonin is proposing is when you actually look at biological meaning in a vertical approach, like he's been talking about, you actually see a lot of consistency among viruses, bacteria, these simple single cell or even non-cellular organisms, and that the density of biological meaning drops off dramatically as the organism becomes more sophisticated.
So is the genome size axis, is that only relevant to the density, or is that relevant to both lines, I guess, both functions?
I think it would be both to some extent. Well,
Yeah.
not that much actually, but a little bit.
Well, the description says information density depending on genome size, and I was just wondering because I know like Arabidopsis, I think that's like broccoli or something, or like
No, it's
cabbage plants.
Thale cress.
Yeah, but I know like plants are known for like huge amounts of polyploidy, so like that their genome size is probably substantially larger, and I'm assuming it's only talking about like haploid genomes. But
yeah, and Arabidopsis is 10. So
where's the blue number 9? The number 9 that's blue, it looks like it's actually in the exact same spot as the red square.
Yeah, it is.
Yeah. Oh,
That's
okay.
interesting.
Yeah.
Huh, I didn't even notice that.
Yeah,
Pointing
that
out
one
about
is.
plants. That's
yeah, plants have
that
crazy
would be
big
something.
genome sizes. But
That's Arabidopsis. So, but it's amazing how similar the human, the fruit fly, and the thale cress are as far as how far up they are in this graph. Whereas
for the blue diamond,
right?
That's,
As far
for total information,
right. That in itself is very interesting.
But as you get up into more complex organisms, the actual meaningful information drops out as a function of the size.
Yeah, and that's where we have the red line.
That's pretty interesting.
So let's take a look at talk about the red line. Yeah, the squares.
Yeah, so that's Koonin's equation that he's describing above that we've been talking about, looking at entropy, but ultimately quantifying biological meaning. You would think humans would have the most meaningful genomes, if there was some kind of purpose or plan at play, but it's the opposite trend.
Or the densest, the maximum information density. So as organisms get more complex, it seems that we can withstand a little bit more in the way of mutation and entropy because of the lower density, basically.
So if you want to move into fuzzy meaning, if you want to scroll back up and read some of that stuff out, Speed.
Yep. The now well-established phenomenon of pervasive transcription that has triggered the infamous debate around the results of the ENCODE project. When pitted against the formal considerations outlined above, suggests a radical line of thinking on the nature of biological information and meaning. The indisputable findings that nearly all sequences and complex genomes, such as humans, are transcribed at some level, at least in some cell types and at some life stages, most likely fit the same population genetic perspective. Conceivably, transcription is pervasive because the selection against spurious promoters and enhancers is not sustainable in small populations subject to drift. Somebody want to comment on that, or want to tear that apart a little bit?
Uh, so this is really interesting to me, and it's something. So I'm involved in human research, human health, medical research, and it was apparent to me how I described the genome as leaky. How all of these areas of the genome seem, you could pretty much find transcripts for just about any region of the genome. Like he said, in some cell types and at some life stages. Uh, but what Koonin is saying here is that you're less likely to see that in these small, efficient, simplified genomes that we would see in viruses and bacteria.
Yeah.
That's interesting. I
Something
didn't
else-
realize that. I guess I kind of had an assumption that all genomes were equally leaky or comparably leaky. Uh, but it sounds like what Koonin is saying is that that's actually a side effect of being a sophisticated multicellular organism. Uh, Leakiness, you know, couldn't couldn't be selected out.
Yep.
If you don't mind, keep reading, because at the beginning of these sections, he's speaking really generally, and I think it's pretty helpful. Don't you think so, Walker?
Yeah, I think it's really interesting. I feel like at least part of his motivation in this paper was sort of a comeback against all of the creationists who have like sort of hijacked information theory, or at least maybe that's how I'm seeing it, because like I'm involved with this. But it seems like him bringing up ENCODE, or his statement about how prokaryotes and more basal organisms have like higher genomic density and are better designed, like the language better designed, to me sounds like he's actually addressing creationist arguments specifically.
Yeah, that's a really good point. I didn't pick up on that, and I think the reason I didn't pick up on that is because I had that feeling every time Koonin was quoted, and then I went to the actual source, and it's either unrelated or the polar opposite of what the creationists construes it to be.
Mm-hmm.
But yeah, to make that provocative statement and actually include the word design, yeah, I think you could be right about that.
Yeah.
Speed, you want me to read a little bit?
Hang on a second here. As long as we're talking about Koonin,
Sure.
and the creationists quoting Koonin, their favorite is to quote his The Logic of Chance, which is a book he wrote, pretty thick book. And the favorite quote is, "Despite many interesting results to its credit, when judged by the straightforward criterion of reaching or even approaching the ultimate goal, the origin of life field is a failure." But then he goes on to talk about all the different ways in which it's not a failure. Not everything is bleak, and there's more to that paragraph. And they love to quote that particular thing. But it should be noted that at the very end of this book, he has another quote in the end notes, which is, "The phrase irreducible complexity was coined by Michael Behe, one of the chief advocates of the anti-evolutionary intelligent design ID concept, in his famous Darwin's Black Box." And then he goes on to say, "To Behe and other ID advocates, the irreducibility of complex biological structures is evidence, even proof, of the inevitability of ID." And then he says, "Of course, ID is malicious nonsense." but the term...
So they've been on his radar for a while.
Right, but the term irreducible complexity is quite evocative. However, evolutionary biologists might prefer to speak of apparent or purported irreducibility of complex structures. So anyway, that's his fire back. That's what he has to say about the intelligent design people. So they should always include his latter quote with the prior quote if they want to be fair and honest.
Speaking of kind of the cherry-picked quotes, there's a Bernhardt paper, I think 2010 or maybe 12, and the title is something like "The RNA World Hypothesis: The Worst Theory of Early Evolution of Life." If you stop there, you would think that the paper is attempting to show the flaws within it. But the rest of the title is, it's the worst theory of the early evolution of life, except for all the other ones. It's kind of what is it? A Thomas Jefferson quote. I may be misquoting, but basically, democracy is the worst form of government, except for all the others.
Right.
Yeah.
I have that paper in the stack back here somewhere, but I'm not going to look for it. So, okay. So, however, in the context of the above formalization of biological information, would it be appropriate to view most of the sequences in the complex genomes with extremely low biological information density as being endowed with fuzzy meaning? So, we're looking at the human genome, which is low in biological density. And we're looking at the ENCODE project where you have all of this spurious transcription. Operationally, sequences with fuzzy meaning can be defined as those that cannot be aligned between genomes that have diverged beyond the threshold of sequence conservation. So when you look across genomes, you'll find some sequences that are just highly identical, and you'll find others that tend to vary quite a bit. So anyway, that's fine. That is due to common ancestry alone. The threshold of sequence conservation that is due to common ancestry alone after the synonymous sites in protein coding genes have reached saturation. This is a conservative definition because it assumes neutrality of the synonymous sites that in actuality are subject to selection, albeit substantially weaker than that of affecting non-synonymous sites. Mm-hmm. So in a lot of these transcribed sites, you're going to see weak selection. In other words, we can get away with a lot.
Huh.
Yeah.
That's
The synonymous sites are subject to weak selection compared to non-synonymous sites. Is that talking about like codon bias?
No, it's talking about more than that. That there are that some of these things that are transcribed don't seem to kill the organism. They're weakly selected for. They might just end up making your face look slightly differently or have no effect at all.
Oh yeah, just like genetic drift type stuff.
Yeah, do you want to straighten me out on that a little bit, Molecular?
I am chewing on the ideas as well, but I think you're on the right track as far as I know. That effectively, kind of going back to the vertical versus horizontal, I think he's looking at sequences that maybe wouldn't conventionally be thought to have a lot of meaning by either horizontal or vertical.
Right.
May still have fuzzy meaning. And I checked, it was Winston Churchill, so I butchered that quote. It wasn't Jefferson at all. So I had to correct the record. So not only are we looking at homology as areas rich with biological meaning among species, but areas that aren't necessarily homologous or not necessarily coding can have this fuzzy meaning. Is that what you're saying, Speed?
Yeah. Let's see. First, the sequences with fuzzy meaning from the material basis of plasticity, from which functional molecules, primarily but not exclusively, regulators of various processes, are continuously recruited to assume better defined meaning. So continuously recruited. So this is where he's talking about. This is where he is talking about how that. Basically, Kimura's neutral theory that massive amount of non-coding, non-conserved DNA can assume better defined meaning over evolutionary time, and that's what
it's the substrate. It's what the creationists deny exists. It's the material basis of plasticity, as Koonin puts it. Or basically, it's where new genes are derived.
Yeah.
It's the foundation from which new genes are derived.
and new regulatory functions, and hence new phenotypes and new pathways, new biochemical pathways. Like I have, and of course it's just my hypothesis, there are a number of these, all of this transcription, they all enter into some biochemical situations, and not many of them are meaningful. They're only the ones that are really, really important to the cell. But still, every now and then, they will form a network of pathways and create a brand new biochemical pathway. Like if you look at, there's something called GPCRs, G protein coupled receptors.
Yep.
And G protein coupled receptors have a 7 alpha helices that go through the membrane, and those are pretty highly conserved, but not as much as what is inside of the cell. But on the outside of the cell, they have all these little loops that lock into things. And if perhaps somebody came up with a mutation that locked into a particular chemical in a flower, then that person would have the ability to smell a different kind of flower. And that would, if that becomes important to the organism, like if that flower, the people or the organisms that smelled that flower got more food, then that would start to be selected for, and you would have an addition of information through de novo gene mutation. Basically, a GPCR would be duplicated. It would mutate. It would lock into a new phenotype, an organism or a bee or whatever that could smell a different kind of flower, and that thing would survive more and hence be selected for. Yeah. So that's a really good example. GPCRs, we have, I think, 700 families or something.
Yeah, that whole concept was one of the things that stuck out to me the most about like intro bio, because we had to, memorize like a couple of different cellular pathways and stuff. And like 2 or 3 of them were G protein coupled receptors as well. Yeah. And to me, it was interesting to see where the pathways were similar, but where they also diverged, right?
Yep.
So you would have like slightly different G proteins, and like one of the G proteins would like auto-catalyze itself, and then others like would rely on like CAMP or something like that, And it was just cool to see like how these different pathways like could have diverged from each other, if that makes sense.
Yeah, when you start to look into the details of the biochemistry, you can see evolution inside of these different pathways, across different pathways, and that's just one of the more stunning ones. So the GPCRs are how we smell things too.
They're essential for tons of cellular signaling in development, in normal homeostasis, wound healing. I mean, it's a ton. Before you move on, he actually said there were 2 levels, and I'll read the second one.
Okay.
So this is the genomic regions with fuzzy meaning. They have meaning. Their meaning can be rationalized on at least 2 levels. So the first level was that these fuzzy meaning regions can be the material from which new genes can arise. And secondly, what he says is that it's not necessarily the transcripts. It's not necessarily that their transcripts are coded into some new gene to provide a new phenotype, like you were describing with smells. But the second level of rationalized meaning that can be found in these regions is the transcription itself can have a function. And reading this, I think what he's referring to are long non-coding RNAs, so that chromosomal structure is at least partly maintained by expression of long non-coding RNAs, and they effectively, if a heterochromatic or a very densely packed region of the chromosome, like the telomeres, if it loosens up for some reason, which this happens, everything's in equilibrium, so it happens. If they loosen up enough and these transcription factors combined, even if they are binding with low affinity, and you have very low levels of transcription, like might be detected by the ENCODE project, what they do is they produce transcripts that instead of being translated, they're shunted to a different set of protein complexes that basically target those complexes directly back to where the loosened chromatin is located, so that you have effectively a signal that says, hey, I have decondensed, I have converted from heterochromatin to euchromatin, bring in all the histone methyltransferases, bring in the DNA methyltransferases, bring in all the necessary equipment, and it converts the loosely packaged chromatin back into heterochromatin. So it's a redundant way to maintain chromatin structure.
Yeah.
So the fuzzy meaning in this sense, the actual meaning, biological meaning, could be, improved chromosomal segregation during mitosis, improved genomic stability over time, which would lead to, decreased chances or risk for cancer, and other elements. It may improve the length of your lifespan. So even though they're not coding a gene or providing a substrate for new genes to be, created from, they still have a valid biological meaning, a fuzzy but valid meaning.
Yeah, there's another important sentence here. The information flow between the domains of defined meaning and fuzzy meaning certainly is a two-way street. Loss of meaning continuously occurs as well in the process of pseudogenization. How would you say that one?
That's a new one to me.
Yeah, creation of pseudogenes. There's no good way to say that word. They should never have created that word. But anyway.
Here, let me define it for all of our benefit. Well, right. Okay, so I looked up that term, and it takes me. It's basically the formation of pseudogenes, which makes sense now.
Retirement gene.
They're non-functional, loci that have some of sequence homology to fully functional genes. Mm-hmm. We know of a pseudogene. We've talked about it quite a bit. DDX11L2 is a pseudogene.
But that that was created by a fragmentation process. I think what he's suggesting here is that there's a process where some genes lose meaning, and you put them on you end up putting them in the old folks' home.
That makes sense. The genome is extremely dynamic and subject to selection. So, in addition to new genes arising, you have genes, being lost over time. Maybe not necessarily selected against. But
heterochromatin and euchromatin is probably deserves a comment. Heterochromatin is the stuff that's packed up sort of in the attic and not particularly being used in the cell right now. And then it becomes unpacked and becomes euchromatin. Is that a fair way to put that?
Yeah.
May go up in the attic.
I could talk about it for hours. So, so if you picture images of chromosomes, if you ever Google chromosome, you see usually they show these X-shaped kind of rounded structures. Mm-hmm. That's a metaphase chromosome. So during the cell cycle, after the chromosomes have been replicated, so you have 2 copies each of the normal complement of chromosomes. But before they're segregated into daughter cells, they're compacted into their most dense form during the entire cell cycle. So when people talk about euchromatin and heterochromatin, they're talking about kind of a steady state, not necessarily the cell cycle. I'm pointing at metaphase chromosomes as kind of an example of how dense chromosomes can be compacted. The opposite. So during the growth phase of the cell cycle, during the phase that most of our cells are at in our body, the chromatin is really loose. So if you were to stain it and look at it, through a microscope, it would look, very diffuse, thread-like, and spread out amongst, within the nucleus. So that's more akin to euchromatin, or transcriptionally active. But basically, heterochromatin versus euchromatin has to do with the amount of coiling and supercoiling, and is largely associated with regulation of gene expression. But it also is associated with DNA replication as well, and DNA repair.
And you had a straw analogy?
Oh yeah. I almost brought that up, but I didn't know if it would be appropriate here. So I was telling somebody, me and Speed were talking about this the other day, that if you were ever, I remember in junior high, in the cafeteria, we would get straws with the paper, lining on the outside, and if you push the paper on the outside down so that it compacts itself all the way into, like a little worm tube type thing. That kind of represents the most compact form of chromatin. And what we would always do, and you guys may have done this as well, if you drop like water onto that tube, it starts to relax and expand. So you have the straw paper, it starts out, maybe 8 inches long. I don't know what the average length of a straw is, but you compact it down to 1 inch. That's like converting euchromatin to heterochromatin. But then you drop water on it, and it starts, the entire thing doesn't start to loosen up all at once, just the region where you drop the water. So you'll have, a little bit of loosening, decompaction, and if you wet the whole thing, it kind of loosens up most of the way. That's how epigenetics and histone modifications and other chromatin modifying complexes, that's what they do to the actual DNA. So if a gene is compacted, it's not going to be expressed. Transcription factors can't bind to the promoter site. RNA polymerase can't, transcribe the open reading frame. None of that stuff is available.
Yeah, and only a damn scientist who works in the lab would worry about the average length of a straw in this conversation.
Yeah.
Anyway,
so let me say one more thing. So the kind of big picture, talking about chromatin and function, we all start out as a zygote. So when the sperm, fuses with the egg and produces that first cell, that's called the zygote. At that point, it divides into two, and those two cells give rise to all the cells in your body.
Mm-hmm.
As it divides, as the zygote is developing into a fetus, different cell lineages begin to differentiate into specific tissue types or tissue precursors. The way that a skin cell maintains its skin cellness, so a keratinocyte, the difference between a keratinocyte and a liver cell, all the same genetic information is there, but in a liver cell, all of the genes associated with being a skin cell are heterochromatic. They're all tightly compacted, they're silent, but in the reverse, in the keratinocyte, in the skin cell, all of the liver cell associated genes are tightly packed. So this packaging of chromatin is largely responsible for defining cellular identity.
Mm-hmm.
And actually, my particular expertise is in molecular oncology, cancer research. one of the hallmarks of cancer is that the fully differentiated normal cell transforms, and one of the characteristics it gains is it begins to lose that fully differentiated identity, and it starts to become more stem-like, and that leads to a huge other conversation about transformation and progression, which we could talk about at some point.
My analogy for heterochromatin and euchromatin is that the heterochromatin is like when I store the Christmas decorations in the attic because I don't need them, but then when it's Christmas and I need them, I unpack them and bring them down, and now they become euchromatin and they get expressed on my Christmas tree. So anyway,
that's functionally that's a perfect analogy.
Yeah, we need to get molecular alchemy to debate either standing or raw mat on like epigenetic regulation and its function in evolution.
Oh yes,
because that would be.
I haven't heard them talk about that.
Really, that's their entire argument.
That's a big one.
Yeah, I've heard them use the word epigenetics, but never in a context
that's correct.
where I would have used it, I guess.
Oh, they claim it's responsible for all speciation after the arc.
Yeah.
That epigenetics did it all, which I think is a real misuse of epigenetics.
So basically their whole idea is that recombination in epigenetics is what explains essentially all diversity in life, especially like within species and within genera, that kind of stuff.
So I mean, I'm an epigeneticist, and I don't want to undercut the value of epigenetics, but there's not a direct link between the epigenetic state of a chromosome and the allelic content of that chromosome. Not that I'm aware of.
That's where they are just plain wrong. I mean, they misuse epigenetics now and they also claim, like Jensen in his book, he actually says epigenetics is a very new field. Like we just discovered it a few years ago.
Like 20 years ago.
Or a few years before he wrote his Darwin's book.
Yeah.
So, and it's been around for quite a while. It's helps.
A decade ago, and I think maybe you and I, Steve, we may have talked about this before, but like a decade ago, everybody in research in molecular biology, if you weren't asking questions about epigenetics before then, you started to. It was certainly a buzzword. For instance, you mentioned recombination. There's a guy in our department who's a world authority on recombination, and he came to our group because we do proteomics and histone epigenetics, and effectively, he's used a model organism for decades to show what recombination is and the steps that occur in order for it to happen, and all kinds of feedback loops for regulation. He's discovered a lot of really cool stuff around around recombination, but he basically started over at square one with all of these hotspots, these sites of recombination, and his interest is look at the histone code at those sites, look at DNA methylation at those sites. Mm-hmm. So it did revolutionize the field, but I have never. I'll have to investigate the literature for epigenetic alterations driving speciation. That seems like a contradiction to me.
I just haven't reached it.
That would get into kind of Lamarckism and
well, how does it survive across multiple generations? I mean, I think in some fungi they found out that it actually goes up to like 100 generations, but typically it's about 3.
Well, it depends on the organism and it depends on the epigenetic process. But the epigenetic state, meaning which genes are turned on, which genes are turned off, that can be inherited. I guess in that way it can certainly influence evolution. I don't know.
Yep.
I'll have to investigate it before I
Yeah.
chat with them.
But anyway, Jensen in 2017 claims that it's a brand new science and it turned evolution on its ear, sort of is what they keep saying, that we just discovered epigenetics and it turned evolution on its ear.
He may have just discovered it.
He just discovered, right?
Yeah.
And that's the thing, when they just discover something, they think everybody discovered it with them. I don't know what that's called, what kind of egocentric bullshit that is. So let's see, we're here, the boundary between, and I think we're going on to another paragraph. What else do we have here to highlight in this? I'm not sure where you were reading from.
Honestly, I think this may be a good stopping point, or somewhere soon.
Okay.
It will certainly. We've certainly said a few provocative things, both from the paper and aside. That's my opinion, though. I'll follow your lead in that respect, Speed.
Well, let's.
Are there any other points of the paper that y'all would like to hit first?
What's that?
Or I said, are there any other high points of the paper y'all would like to hit?
Yeah, there's we're still on fuzzy meaning. The sea of fuzzy meaning sequences. The sea of fuzzy meaning sequences. In the expansive pool of long non-coding RNAs, there are many that are represented by orthologs, even distic organ. Let's see, however, I think he's talking about what drives evolution here. And I'm not sure how much we might want to highlight a little more. I think section 4 is the last section of the paper, isn't it? I'm not entirely sure. Oh, the relevance of the fuzzy meaning for evolution.
Yeah. Oh, yeah. We're almost done, actually. There's just section 4 and then the conclusion.
Yeah. In particular, evolutionary innovations does not appear to be limited to non-coding. Limited to non-coding DNA. Let's see. It has been observed that most of the novel eukaryotic proteins adopt alpha helical folds and seem to have evolved from generic repetitive coiled coil proteins. So he's talking about what they are always asking us for. How did we get from fish to fishermen? And basically this is de novo genes that he's talking about. And he's talking about coding sequences for pre-proteins. And some of this stuff could be expanded on a little more, but we don't want to dig too deep in this. But that's interesting, coding sequences through a stage of pre-proteins. And this section right here talks about how de novo genes develop and new proteins, new function, right? From that sea of fuzzy meaning, which I love.
That's interesting. That's what I love about these papers from this issue, is they, all of these words, all of these concepts I deal with on a daily basis, but looking at it in this context, I mean, molecular biologists kind of take some of this stuff for granted.
Yeah. And then he says, upon acquiring a specific function, the evolution of these proteins substantially slowed down. Their meaning was sharply defined as they left the fuzzy domain, meaning that they now become conserved for that organism and all of its descendants. So
no longer fuzzy meaning, but specific and detailed meaning, like he was talking about before.
Yep. And the last section, is a way to summarize this, much like in here.
In this section, he kind of extrapolates the idea out more. I think
he drills down.
Yeah.
Okay.
Well, I think you could skip it, or yeah, go ahead. Yeah, let's read it.
Continuous transfer of meaning. Let's say we're kind of skimming over it here. We can't read the entire thing online. Between selfish elements, host is a major evolutionary trend that comes in several guises. So then he has first. So what is first? Genes from the selfish elements are often recruited by host organisms such that The specific activity of the encoded protein is modified and appropriated for host functions. So he's talking about evolution here.
Key examples include the essential eukaryotic enzyme telomerase that is required for linear chromosome replication that was recruited from a bacterial retroelement for its reverse transcriptase activity.
Yep.
And then hedgehogs.
Yeah. Gotta love hedgehog.
Well, these are there's a whole host of genes, sonic, hedgehog,
Indian hedgehog,
that were identified, I think, in Drosophila, that have to do with cellular differentiation, organ development, and even bilateral symmetry. Basically the development of a complex multicellular organism from a single cell.
Yeah, and that's really interesting. When you look at embryology and development, you find that very small changes in some of these non-coding RNAs and in some of these proteins and how they express across a zygote when it's developing, you form these gradients, and these gradients can be separated into bands so that at 20% concentration, this pathway will get turned on. At 40%, another pathway will get turned on. Mm-hmm. So you look from one side of the cell to another, and that effectively creates the organism from the zygote as these different interactive, this big dance of epigenetics takes over and differentiates cells into an organism. But if you make small changes, you can end up with pretty drastic changes in body plans and in what basically the structure of the phenotype. So that is where I became so convinced of evolution when I saw how a small change could make such a massive change in an organism's, how its organs develop and whatever. Like looking at how lungs develop and how tetrapod limbs develop, all of that are rather small changes can make very big differences in the evolution of your cells within the growing organism, basically.
We all start out as single cell organisms.
Yeah, and that's
isn't that kind of amazing?
And how does random chemistry take a single cell and turn it into a human being? I've asked creationists this, and what they look to is, all that information is encoded in the DNA. And it is.
So it's
not. It is to an extent, but it's also.
But it's encoded in the DNA after millennia of selection and evolution.
Right, and it stays encoded in the DNA, but it's also encoded in the zygote that comes from the mother and the father, the fertilizer. It's encoded in that first cell, the zygote has proteins that come from the mother's DNA, and so very little from the father. And so there's some information that's passed from generation to generation by the zygote, by the cell, not by the DNA. And then there's other information that comes from the environment that the organism grows in. So there's a little more than just, it's not all cleverly encoded in the DNA. It's a combination of environment, of the ancestors, and the DNA all together. And it's sort of like this evolving dance that keeps rolling over and changing a little bit every time and adapting to whatever the environment is. And that is the beauty of morphogenesis in evolution. So anyway.
All right. You want to kind of wrap this up? Maybe.
Yeah. Let's see what his conclusion is. Are you, do you need to get out?
No, not really.
Okay.
I'm keeping, so this kind of approach, the paper trails approach.
Yep.
We've been going an hour and 20 minutes. Previously I had suggested to you that we just show the figures and just kind of talk about it.
Right.
I do like having the words up so that, people following along could see, there's much more to read there. And what do you think, Walker, as far as like as a video series?
Well, we can talk about this after we finish wrapping this part up. Do you want to?
That's a good point.
Yeah.
Okay, for the conclusion, we just need to go. And the other thing that we could consider is having edited 2 different versions of the same talk.
Yeah, like a long form version and then like a cut down.
That's a really good idea.
Yeah, after we edit and produce this version, we can come up with a condensed version where one of us
you may be able to just edit a condensed version out,
Yeah,
some of our tangents and.
Okay, so what do we need from this? I'm trying to figure out what I highlighted in my conclusion. Of course, I have it on the paper, and I did not put it.
Scroll down a little bit more so we can see the figure.
Okay, I was looking at this first. I highlighted the biologically relevant information is more akin to meaning than to entropy. So I'm not sure what he was going with. It's type of information. Well, let's look at the figure. You can talk about that. Do you want to?
This particular figure goes with the section 4 where he talks about selfish elements, but we didn't really we didn't even define what a selfish element was.
Okay.
I mean, we can potentially go back to do that.
Would that be a highly conserved region, a selfish element?
I don't want to paraphrase and get it wrong.
Okay, let's.
It was in 4 back.
Well, let's see.
Is that the whole thing of like Dawkins' selfish gene idea, where like certain regions of the genome are going to be conserved just because they have a predisposition for being conserved?
I think that may be the principle.
Okay, the sequences of innumerable selfish mobile genetic elements that are integrated into the genomes represent the majority of genome sequences in animals and plants are generally meaningless for the host organisms. Okay,
for most of those elements.
Yeah, he's referring to the Dawkins selfish gene idea.
Yeah.
And then
he doesn't really define it in his paper. I've searched through all the selfish elements parts.
Yeah, he just sort of, let's see, from the selfish elements point of view, he's referring to the parts that are not essential to the organism, but yet these things have meaning to themselves.
Well, so the whole idea with that is that certain regions of the genome are going to just naturally be better conserved. It's basically like Darwinian fitness on a genetic level. Yeah. Right. So you're going to have a certain region of the genome that's really good at making sure it's conserved. And if it's good at making sure that region is conserved, then that will be passed on. Right. Yep. And if that's passed on, then all of its descendants will also have that same highly conserved, but not necessarily functional region of the genome. So that's what it means by, at least that's what Dawkins means by selfish genes.
Yeah. They're talking about the viruses, retrotransposons or transposons, host genes involved in defense and adopt them for counter defense. Thus, the sequences have been meaningful for selfish elements become meaningful for the host, and vice versa. So, vice versa. That's interesting.
Yeah.
So, okay. So we're back down to our figure. So we have, loss of meaning, transfer of meaning from selfish genes to the host gene pool. And then we have fuzzy meaning, and I don't know.
I think the biggest concept that the three of us, can take from this is the idea that traditional information theory as a measure of Shannon entropy is not an appropriate metric to look at genetic information.
Right.
That instead, derivations of the Shannon entropy equation that take into account, the actual relevant contributors of evolution, biological function, uh, the laws of physics surrounding the activities of a cell. That ultimately, that's a much better metric, and it gives you what Koonin is calling a measure of meaning, biological meaning. And the second half of this paper is basically Koonin showing that if you use that approach, if you look at it vertically and not horizontally, if you look for biological meaning and not simply quantify the total amount of information, that you can actually see meaning that is consistent with these other fields like evolutionary biology, these fuzzy sections of the genome that don't necessarily code for a functional protein. I think really the fuzzy meaning section and kind of the selfish gene section is just further support for the argument that we should be talking about biological meaning, not biological information.
Yeah, and thus the famous dictum: nothing in biology makes sense except in the light of evolution, which is why I went back to that.
That was perfect.
Yep. So
if
nowhere
you
in
want
here.
to end it there, and we have a conversation, the three of us.
Yeah, we can, but nowhere in here. Well, this is all offline anyway, so
I got you.
we've got to edit it so we can have a conversation, keep that recorded in case there's anything meaningful that comes out of it.
Sure.
But the let's see. Well, I was going to say something, and my train of thought went away. It went off the rails. Where did it go?
That's what I was going to ask you though. If you were planning on editing, I don't think it will require a huge amount.
Yeah.
Honestly, though, if you can take a note as far as my preference, I think I could do a better job describing epigenetic mechanisms than I did.
Yeah.
But
Okay.
I kind of I'm here because I trust your creative, skills and
Oh shit! Don't do that. Don't do that.
not just your creative skills, but I don't think you're gonna try and make any of us look bad. You know what I mean?
Right, creative skills I have as far as editing skills and videos. Man, I don't know what the fuck I'm doing. Yeah, but it really is hard to edit these things out. It's very difficult. But if you want to, come on like tomorrow and re-record part of this or script it, or the other thing I can do is I can put these things up to my YouTube site and they transcribe them for me, but I put them up privately.
Oh, nice.
And I get a transcription and we can look at it and we
Yeah.
can start. And that would help for editing greatly. So I could probably do that.
Absolutely, because we could all weigh in on, we could highlight areas we think should be saved or edited out.
I got the train back on the track. Nowhere in here he talks about meaning and basically meaning of biological information. Nowhere in here does he suggest that we need a conscious, intelligent designer or have any reference to anything other than a physical way to measure, find a metric for information. And that is something that we humans tend to do when we're trying to understand things. It has nothing to do with how these things happened. So,
well, you saying that, it occurs to me, I wonder, I'd love to ask Koonin, taking all of this that we've just read and I mean, it's convincing, but now let's talk about abiogenesis. Let's talk about the origin of biological meaning. I'd love to pick his brain on that, because surely the macromolecules that gave rise to the first cells, there must have been meaning. It randomly sequenced oligos of RNA working together enough It would be interesting to think about the origin of meaning in this context.
Yeah, he is only 63 years old, and he is in Russia. His thesis was multi-enzyme organization of myocarditis. Did I say that right? Myocarditis virus replication complexes. 1983 is when he did his thesis. So, and he's written another book which I don't own. Oh my God, I don't own a book. There's a book. Oh no. What are we gonna do about that?
Let's find an electronic version
For free? Yeah.
Yeah. Hey, I do need to wrap it up here. I got kids running around, and
Okay, good enough.
Tonight's my wife's night off, so I can't be on.
Okay, I'll go ahead and try to upload this. It's under 2 hours, and I think I can get a transcript by tomorrow or the next day. And this is going to take some more work, right? And Walker, if you want to, and then if you guys want to come on and record more information that we can, clip in at some point. But,
I gotta say though, I do like this idea for paper trails. I think just how it all ended up sorting out, this may not be the best paper to attack the Brotherhood with,
yeah, but we sort of have, we want to have a repertoire of a lot of papers particularly from that Royal Society, and then we can bring them in for debates, or if anybody else who's debating these guys, we can be a resource for them. Oh, yeah. That's kind of how I view our site, as sort of a resource for debaters. And at some point, some of us are going to go in and do the warrior thing. So,
yeah.
But if we have a pool of this, and if we've all gone through these papers, I've
learned a ton.
Yeah, when asshole comes up with, oh, I was reading a paper, like, okay, which one? And if we happen to know that paper, then we'll say, let's talk about it. Let's drill into it. And what did you get out of it? And he will fall apart, and he'll get pissed off and rage quit or whatever. So that's the way to deal with these people in debates, is we have to have the information almost like R.J. Downard says, you have to, in your opening, you have to defeat everything that they had brought to the table, even though you don't know what it is yet. So that's kind of his method. But Maddox nailed him on Modern Day Debate when he came up with that ribosomal pausing, because RJ had never heard of it, and I had never heard of it. So I had to go look it up, and so for the next 4 days, I was studying ribosomal pausing to figure, because they always come up with something new and they sort of blindside you with it, like fucking satellite DNA on chromosome 2. All of a sudden he starts talking about it, and I'm like, wait a minute, why are you talking about satellite DNA in the telomere fusion site? I was aware of it in the centromere, or the degenerate centromere, but I was not aware of it in telomeres. And then, of course, I found, where they got the information from was goddamn Tompkins. Yeah. Anyway, okay. Well, we can wrap her up. Walker, you want to hang out for a minute?
Sure. Yeah.
I think Immutable wanted to come on and chat with us.
I wish I could stay on for 2 more hours, but I got to go now.
You're good.
Good talking to you guys.
You mean you have a life? Okay, well, go get rid of that.
Later, guys.
Okay. Okay, let's see. I'm going to. You want to hang out for a minute?
Yeah.
Let's see. So I'm still recording this. Oh yeah, I don't want to do that. I want to stop this recording and start a new link. Can you come on in like a minute, or do you want to chat and leave, or what do you want to do? What do you like?
Like, are you saying you want to start like a different call?
Yeah, Immutable wants to come on, and I don't want this one to go too long, so I want to end the broadcast and download it.
That's fine.
So I'll send out a new link.
Yeah, that works.
Okay, I'll do that immediately. Okay.
All right.
See you in a minute.
Yeah, see you.